Welcome to Sir Robert Chiduk Tutorials. Learning is 24/7. Hello and welcome to Sir Robert Chiduk Tutorials. Today we proceed and look at the process of mitosis. The process of mitosis is covered under a process called the cell cycle. So we are going to discover what happens, the processes that happen under the cell cycle. So you'll find that many damaged, dead, and worn out tissues can be replaced by growth and division of other cells. So now you see, the cells have to divide because the cells die. The cells get damaged. Some get damaged, some will die, some become worn out. So these cells have to be replaced by growth of other cells. But these cells, for them to grow, they come from the division of other cells. So this brings the concept of mitosis into play. Most body cells have 46 chromosomes and they divide by mitosis, a process that results into two new genetically identical daughter cells. So this is the process of mitosis. So mitosis is a process whereby two identical daughter cells are formed from a parent cell, right? So a process whereby a parent cell divides to form two identical cells is called mitosis. The only exception to this is the formation of gametes or the sex cells. So mitosis does not happen in the female and male gametes, in the sex cells. In the sex cells or in the gametes, we call it meiosis, not mitosis. There is meiosis. For example, the ova and spermatozoa which take place by meiosis. This I have already said it, right? So you can see that this man is full of information. The period between two cell divisions is known as the cell cycle, which has two phases that can be seen on a light microscope. So this period of cell cycle is characterized by two phases, right? It is known as the cell cycle and it has got two phases that can be seen on the microscope. And these two phases are called the M phase and the interphase. We have two main phases, the M phase and the interphase. So now let us delve and get right straight to the process, to the cell cycle, and discover what happens to the cell cycle. Do you see this diagram? Do you see this diagram clearly? Do you see it? You can see the M phase where cell division is happening, right? This is the cycle. It's even marked with arrows, right? So this diagram is going to illustrate the whole cell cycle, how it happens, right? So let us look at the interphase. So this is interphase. If you check, let me highlight so that you're able to see. Let me highlight so that you see. So here, this is interphase, right? So where is M phase? Where is M phase? M phase is here, where the actual cell division occurs. So you see that the whole green region is interphase, right? And its other subphases. And then this pinkish division is the M phase, right? Let me highlight it so that you are able to see clearly. Here, this is the M phase, right? I think you have seen. Right, now, the interphase, this is the longer phase, and three separate stages are recognized. So the interphase is the longest phase, and it has got three separate stages that are recognized. What are some of these stages? The first stage is called G1, right? The cell grows in size and volume. Let us see the G1. The G1 phase. This is the G1 phase. Let me highlight it so that you're able to see. Let me highlight it so that you see. This one is the G1. Have you seen the G1? That's the G1, this one. It's one of the phases of M phase. We also have S phase, which is yet another, this one. This is the S phase, right? We also have another one, the G2 phase. The G2 phase, let me highlight again so that you're able to see. This one. This is the G2 phase. So we are going to see what happens during those stages. So we say the first gap phase, the first stage under interphase is called the first gap phase, G1. The cell grows in size and volume. So under this stage, the cell is growing in its size and in its volume. This is usually the longest phase and most variable in length. Sometimes cells do not continue around the cell cycle but enter a resting phase instead, which is the G0. Now, synthesis of DNA, which is the S phase. So S phase, this S phase, right? I think you can see it's written DNA replication. S is resembling synthesis. S is for synthesis, right? So it's for DNA. Under this stage, there's DNA synthesis of DNA under S phase. So the chromosomes replicate, forming two identical copies of DNA. Therefore, following the S phase, the cell now has 92 chromosomes. That is enough DNA for 2 cells and is nearly ready to divide by the process of mitosis, as you can see. Now let us look at mitosis. Mitosis, this one is a continuous process which involves distinct stages as seen by the microscope. So let us look at 2 stages under mitosis. So mitosis has got its own stages as well, as we are going to see as well. So I'm going to highlight and show you the stages of mitosis. So mitosis happens here. This is where mitosis happens, right? That's where mitosis happens. And those stages that are there, you can see we've got Prophase, with Metaphase, with Anaphase, we also have Telophase. So let us look at those stages under mitosis. So this is a continuous process involving 4 distinct stages as seen by the light microscope, and the 4 stages we've seen them. That is Prophase. These are the stages, like I indicated to you earlier on. These are the stages. These are the stages of mitosis, with this Prophase, with this Metaphase, with this Anaphase, and with this Telophase. So those are the 4 stages of mitosis, as you can see. So let us look at one of the stages, which is called the Prophase. During this stage, what happens? That's what I want you to understand. During Prophase, this stage, the replicated chromatin becomes tightly coiled and easier to see under the microscope. Each of the original 46 chromosomes called the chromatid at this stage is paired with its copy in a double chromosome unit. The 2 chromatids are joined to each other at the centromere. The mitotic apparatus appears, and this consists of 2 centrioles separated by a mitotic spindle which is formed from microtubules. The centrioles migrate one to each end of the cell and the nuclear envelope disappears. So for you to be able to understand this stage, I want to take you to this diagram which clearly illustrates those stages. This diagram is able to illustrate those stages, right? This diagram illustrates the stages of mitosis, this one. So the first stage here is this prophase here, right? So let us look at the mitotic spindle. Let us look at the nuclear membrane here. You see this nuclear membrane? You see the centromere, right? The centromere, the replicated chromosome, which is the chromatid, is here. These are the centrioles, and this is the mitotic spindle here, right? So what are we saying now under prophase? Okay, what are we saying now under prophase concerning the same? So here we are saying the replicated chromatin, it becomes tightly coiled and it becomes easy to see under microscope. So the chromatid at this stage is paired with its copy in form of a double chromosome unit. And the chromatids are joined to each other at the centromere, like I indicated to you. The mitotic apparatus appears. This consists of two centromeres separated by mitotic spindles. So these are the centrioles. These are the two centrioles, right? That we have been talking about. Now, after this stage of prophase, we have metaphase. What happens under metaphase, right? Because from the previous stage, we have seen that the centrioles migrated one to each end of the cell, and the nuclear envelope disappeared. This is what happens, right? So under metaphase, what is going to happen? The chromatids align on the center of the spindle, attached by their centromeres. So what do you learn here? The chromatids align. So there's alignment of chromatids. There's alignment of chromatids. That is one thing to note. At the center, where does it happen? At the center of the spindle, right? And it is attached by their centromeres. You are going to see it in the diagram as I'm going to show you. So now here is the process of metaphase. The diagram is illustrating metaphase. So it's highlighting metaphase, right? So this is metaphase. Here, what are you seeing? These are the centrioles that you saw, right? There's alignment. There's alignment of chromatids, right? The chromatids are going to be rearranged and aligned right at the center. They are going to be rearranged and aligned right at the center here, right? This is where the chromatids are now. They are right at the center of the mitotic spindle. And the nuclear envelope, now it has disappeared. There is no nucleus. As you can see, the nuclear envelope has disappeared. The chromatids have been rearranged. They've been aligned right at the center of the mitotic spindle. So this is what happens under the process of metaphase. That's what happens under the process of metaphase. Now, what happens under anaphase? The centromeres separate, and one of each pair of sister chromatids, now called the chromosomes again, migrate to each end of the spindle as the microtubules that form the mitotic spindle contract. So let us see what happens under anaphase. What do we learn under this process of anaphase? So there are critical issues that I want you to master, right? So under anaphase, one thing that I want you to know is that there is a separation, right? This is a keyword. There is separation of centromeres. The separation of centromeres, and one of each pair of sister chromatids migrate to each end of the spindle. So there is migration, right? There is migration of the chromosomes to each end of the spindle. So those are the two major issues that happen under anaphase. There is a separation of centromeres, right? And each pair of chromosomes is going to migrate to each end of the spindle. Those are the two major things that happen under anaphase, right? So let me show you here under the diagram that shows the stages of mitosis. Right, do you see anaphase now? So here we are looking at the process of anaphase. This is anaphase, right? There was separation of the centromeres. You see, this one is on its own, this one is on its own. They've separated, right? The sister chromatids, they've separated. So they've migrated now to each mitotic spindle. So the other one is here, the other is here, right? So there was separation, there was migration, there was separation of centromeres, there was migration of sister chromatids or the chromosomes to each pair of the mitotic spindle, which is what is happening here and what is happening here. As simple as that. Now let us look at the last stage of mitosis, which is called telophase. What happens under telophase? So under telophase, the mitotic spindle disappears. So here there's disappearance. Mark this word, there's disappearance. The mitotic spindle disappears. So there's disappearance of the mitotic spindle. The chromosomes uncoil and the nuclear envelope reforms. So the nuclear envelope will reform. That is the second point. The nuclear envelope will reform. So two major points. There is disappearance of the mitotic spindle, number one. Number two, the nuclear envelope is going to reform again. It had disappeared in the previous stage. Now it's going to reappear. Now following telophase, cytokinesis occurs. What is cytokinesis? The cytoplasm, the intracellular organelles and the plasma membrane split forming two identical daughter cells. So what we are going to see here is that there is going to be splitting of the intracellular organelles and plasma membrane. They are going to split now and form two identical cells. So there is cell division here taking place. The organelles of the daughter cells are incomplete at the end of cell division that they develop during interphase, right? So the daughter cells are not yet mature. They are going to mature and develop during interphase, which is another stage different from mitosis. So this is the last stage of mitosis, right? So in summary, what have we learned? In summary, we've seen that the process of mitosis is under a concept called cell cycle, right? Cell cycle. Why is the cell cycle important? Cell cycle is important because some cells die, some cells get damaged, some cells become worn out, and these cells are supposed to be replaced. But how are they going to be replaced? And how are the cells manufactured and produced in the human body? The cells come from parent cells. So the parent cells will divide, right? They'll divide into two identical cells at any given time. And that cell division occurs under a process called mitosis, right? So the whole process is called cell cycle, which happens under two main processes, that is interphase and mitosis. Mitosis has got its own stages within it. That is, we've got anaphase, we've telophase, we've metaphase, right? And then under interphase, we've stages like G1, G2, S phase, which is for synthesis of DNA, and the G1 and G2. And those are the stages of the cell cycle. So this is what we've learned today. Until next time, thank you so much for your time.